There is a building on a Marine Corps base in Quantico, Virginia, that most people have never thought about. It sits inside a federal installation on 547 acres of woodland in Prince William County, roughly 30 miles south of Washington, D.C. You cannot walk in off the street. You cannot file a request to visit. You cannot send your evidence there without going through a formal process that involves law enforcement credentials, chain of custody documentation, and the kind of institutional trust that takes years to build between a local agency and the federal government. Inside that building, approximately 500 scientific experts and special agents work every day on cases from across the United States. Cases sent to them free of charge by state and local law enforcement agencies that do not have the equipment, the expertise, or the methodological capacity to process what they found at their crime scenes. The FBI laboratory at Quantico is not a TV show set. It is not the clean, blue-lit workspace that procedural dramas have spent 30 years making the public think it looks like. It is a working scientific institution, peer-reviewed, court-tested, methodologically conservative in the way that institutions whose work ends up in front of juries have to be. And it is, by every measurable standard, the premier crime laboratory in the United States. Right now, inside that building, there is a hair sample from Nancy Guthrie's home. Not the glove DNA, not the blood from the porch, not the video footage, or the backpack, or the witness tips. A hair, physically small enough to fit between two fingers, forensically enormous enough that the most qualified DNA expert in the public discussion of this case, CeCe Moore, the genetic genealogist who helped the FBI work on the Golden State Killer case, called it a potential Hail Mary, a last resort, a thing you throw when everything else has reached its limit, a thing that, if it connects, changes everything. Tonight we are going inside the Hail Mary, not the politics, not the timeline, not the institutional conflict we have already covered. Tonight is just the science. What that hair actually is, what it contains, what the FBI's laboratory at Quantico can extract from it that a private lab in Florida cannot. What those extractions can tell investigators about the person who left it inside Nancy Guthrie's home. And why the specific combination of capabilities available at Quantico applied to this specific type of evidence represents the most sophisticated biological hunt in the history of this investigation. By the time this is over, you are going to understand why a single hair, smaller than a matchstick, lighter than a feather, easily overlooked by anyone not trained to see it, might be the object that finally gives this case a name. Let's start at the most basic level. Because the public understanding of hair as evidence is shaped almost entirely by what television has taught, and what television has taught is wrong in ways that matter enormously here. The first thing to understand is that hair is not one thing forensically. It is a collection of biological systems layered on top of each other, and each layer speaks a different forensic language. The outer layer, the cuticle, is a series of overlapping scales, like roof shingles. They can tell an examiner about species of origin, body location, whether the hair was forcibly removed or naturally shed, and whether it has been chemically treated, dyed, bleached, or permed. Those determinations are made through microscopy, physical examination under magnification, and they are the first step in any hair analysis because they narrow the field before the laboratory even begins molecular testing. The middle layer, the cortex, contains the pigment and the structural proteins that determine color, texture, and diameter. The cortex can tell an examiner about the racial origin of the donor, not with absolute certainty, but with probabilistic strength, and about the donor's age, because hair structure changes across a human lifespan in ways that are measurable under a comparison microscope. The innermost layer, the medulla, is a canal that runs through the center of some hairs but not all. Its presence, absence, pattern, and interruption pattern are additional physical characteristics that can contribute to comparative analysis. None of that is DNA. All of that is physical morphology, the shape, structure, and composition of the hair itself. And it is the forensic baseline that every hair examination at Quantico begins with before a single molecule of DNA is extracted. It tells investigators whether the hair is worth pursuing further. It tells them what they are dealing with before they open the molecular chapter. In the Nancy Guthrie case, the hair from the home was described as rootless, meaning it was a shed hair rather than a pulled hair, and it did not have a follicular tag, the small cluster of cellular material at the base of a rooted hair that is the richest source of nuclear DNA. That designation, rootless, is the one that generated the most public concern when it entered the coverage, because for decades, the conventional forensic wisdom was that rootless hair was a limited source of DNA, that without the follicle, you were looking at mitochondrial DNA at best, and mitochondrial DNA, while useful, could not produce the individual identification that nuclear DNA provides. That conventional wisdom was true in 2010. It was becoming outdated by 2018. And by 2026, it has been comprehensively dismantled by research, by laboratory development, and by the specific, documented, case-closing success of techniques that were not available when most of the people commenting on this case formed their understanding of what rootless hair can and cannot do. Let's talk about the science that dismantled the old wisdom, because this is the part nobody is explaining clearly, and it is the part that makes the Quantico hair sample so significant. The conventional belief that rootless hair contains only mitochondrial DNA was based on the chemistry of hair formation. As a hair grows, the cells in the shaft die. Living cells contain nuclear DNA, the full 3.2 billion base pairs that constitute the human genome, the profile that is unique to each individual. Dead cells lose their organelles, including the nucleus. So as the hair shaft forms from living follicle cells, the nuclear DNA that was present in those cells degrades. What remains in the hair shaft is mitochondrial DNA, which is found not in the nucleus but in the mitochondria, small cellular structures that exist in far greater numbers per cell than the single nucleus. Mitochondria survive the cell death process better than nuclei, which is why mitochondrial DNA is recoverable from rootless hair when nuclear DNA is not. But here's what the newer research revealed. The nuclear DNA in shed hair does not completely disappear. It degrades, it fragments. It becomes highly degraded, short-strand material that is difficult or impossible to analyze using the standard short tandem repeat, or STR, methodology that CODIS relies on. STR analysis requires intact, sufficiently long DNA fragments to work properly. Highly degraded, short-strand nuclear DNA fails STR analysis, which is why the standard laboratory approach to rootless hair has historically been to skip nuclear DNA and go straight to mitochondrial. But STR analysis is not the only tool, and that is where the revolution begins. In 2018, a biomolecular engineer named Dr. Ed Green at UC Santa Cruz, working with genetic genealogist Barbara Ray Venter, the same scientist who helped identify the Golden State Killer, developed a technique that takes the degraded, fragmented, short-strand nuclear DNA present in rootless hair and sequences it using whole genome sequencing methodology rather than STR analysis. Instead of looking for specific known locations in the genome to produce a profile, the whole genome approach sequences everything, all the fragments, all the degraded material, and then computationally reconstructs a genotype from the coverage produced. The critical threshold, established by National Institute of Justice Research, is approximately 300 million sequence reads from a 5-centimeter hair sample, which produces approximately one-fold average genome coverage. One-fold coverage means every location in the genome was sequenced at least once on average. That is not the deep coverage of a research genomics project. Those typically aim for 30-fold or higher. But for forensic genetic genealogy purposes, one-fold coverage is enough. Enough to accurately infer genotypes across the genome. Enough to produce a genomic profile that can be uploaded to a consumer genealogy database and compared against the profiles of biological relatives. From a rootless hair, a hair without a follicle, a hair that standard forensic methodology would have assessed as limited for nuclear DNA and sent to mitochondrial analysis. That is the revolution. And the company that operationalized it for forensic casework, taking Green's technique from a university lab and turning it into a deployable forensic service, is Astrea Forensics. Let's talk about Astrea Forensics, because this is the specific company that exists at the intersection of the rootless hair evidence in the Nancy Guthrie case and the genealogical methodology that solved the Golden State Killer. And most of the public commentary about this case has not connected those dots clearly. Astrea Forensics was founded specifically to apply ancient DNA techniques, the same methodological framework that scientists use to sequence DNA from woolly mammoths, from Neanderthal bones tens of thousands of years old, from Egyptian mummy tissue, to modern forensic samples. The logic of that connection is powerful once you understand it. Ancient DNA recovery techniques were developed to extract genomic information from samples that are so degraded that standard laboratory methods fail completely. The challenges of ancient DNA, fragmentation, degradation, low quantities, contamination, are the same challenges that rootless hair presents in a forensic context. The UC Santa Cruz team, which Dr. Green leads, solved a 40-year-old murder case, the 1982 rape and murder of 9-year-old Darylyn Johnson in Idaho, using rootless hair recovered from the original crime scene. The hair had been in an evidence freezer for decades. Standard STR analysis had failed repeatedly across 40 years of periodic case review. When the sample was submitted to the whole genome sequencing approach, it produced a genomic profile. That profile was uploaded to a genealogy database. A family tree was constructed. A candidate was identified, David Allen Dalrymple. A discarded DNA sample confirmed the match. Dalrymple was convicted in June 2024 and sentenced to 2 consecutive life terms without parole. 40 years, a rootless hair, whole genome sequencing, a genealogy database, a family tree, a discard sample, a name, a conviction. That is not a theoretical pathway. It is a documented, court-proven, jury-confirmed pathway that went from evidence that standard forensics could not crack to a life sentence. And the hair sitting at Quantico in the Nancy Guthrie case is the same category of evidence. Rootless, complex, challenging by standard methods, that the Darylyn Johnson case hair was before the right technology was applied to it. The question the public has been asking is whether the hair from Nancy Guthrie's home can be analyzed the same way. And the answer, based on the specific capabilities now deployed in this investigation, is more optimistic than the public coverage has suggested. Here is where the Nancy Guthrie case gets specific, because the hair is not just rootless, it is part of a complex mixture. CeCe Moore's assessment was Nancy's DNA plus 2 or more unknowns. And that complexity is what makes this evidence both the most powerful and the most technically demanding piece of the forensic puzzle. A complex mixture, in forensic DNA language, means a sample that contains the biological material of more than 2 people. Standard STR mixture analysis gets unreliable above 3 contributors. The interpretation becomes statistically ambiguous because the number of possible genotype combinations increases exponentially with each additional contributor. A 2-person mixture has clear majority and minor contributor profiles. A 3-person mixture is harder. 4 or more people and standard methods begin to struggle with the assignment of specific alleles to specific individuals. That is the challenge the private Florida lab encountered. That is why the hair needed to go to Quantico and potentially to the advanced mixture deconvolution software that CeCe Moore said had been expedited specifically because of this case. What does mixture deconvolution actually do? In the simplest terms, it takes a sample that looks like a biological tangle, multiple genomes layered on top of each other in a single test tube, and computationally separates the individual threads. It assigns specific genetic variants to specific contributors, distinguishing which alleles belong to Nancy, which belong to one unknown, and which belong to others. Done correctly on a sufficiently characterized sample, mixture deconvolution can produce individual genotype profiles from contributors who were identified only as unknown before the analysis. Those individual profiles, separated from the mixture by computational analysis, are then what gets uploaded to genealogy databases, not the mixture as a whole. The deconvoluted individual profile of each unknown contributor, and each unknown profile uploaded separately generates its own set of genealogical matches, its own family tree, its own candidate identification pathway. So the complex mixture, rather than being a forensic obstacle, is potentially a forensic multiplier. If the deconvolution successfully separates 2 unknown profiles from Nancy's known profile, investigators have not 1 genealogical trail to follow, but 2. 2 unknown males. Remember, more than 1 suspect was officially described as not ruled out. With 2 separate family trees being built simultaneously, 2 separate candidate identification processes running in parallel, 2 separate discard sample surveillance operations potentially closing in on 2 separate people at the same time. That is not the single glove, single unknown profile picture that defined the public understanding of the DNA evidence in this case for the first 100 days. That is a fundamentally different investigative landscape, richer, more complex, more technically demanding, and potentially more decisive. Now let's talk about what else Quantico can extract from that hair, beyond a genealogical profile. because the DNA is the headline, but it is not the only story the hair tells. Physical appearance prediction. The FBI's laboratory, using next-generation sequencing data from a hair sample, can derive probabilistic predictions about the physical appearance of the donor, not from the DNA profile used for genealogy, but from the same genomic sequencing data applied to a different analytical layer. Skin color, eye color, hair color, ancestry, and biogeographical origin. These are phenotypic predictions, estimates of what the person looks like based on which genetic variants they carry, and they are probabilistic rather than definitive. They cannot tell you with certainty that the suspect has brown eyes, but they can tell you there is a high probability of brown eyes based on the specific combination of genetic variants in the sample. And in a case where the only visual evidence of the suspect is a masked, gloved figure on a doorbell camera, even probabilistic phenotypic predictions can narrow the field significantly. The FBI's laboratory has published on the validation of next-generation sequencing for their forensic casework, including a specific validation study using the PowerSeq CRM nested system, published by the National Institute of Standards and Technology. They have formally integrated NGS into operational casework. This is not experimental technology being tried for the first time on the Nancy Guthrie evidence. It is validated, peer-reviewed, court-tested methodology that has been formally accepted into the FBI lab's operational pipeline. Beyond phenotyping, the NGS data can also be used for ancestry informative markers, genetic variants that carry information about the geographic origins of a person's ancestors. Ancestry informative marker analysis, combined with the family tree work of genealogical comparison, can further constrain the candidate pool by specifying not just who the biological relatives are, but where those relatives' family histories originate geographically. If the suspect's genealogy tree shows heavy weighting toward a specific region, that geographic information layers into the investigative picture alongside the physical profile and the family tree. And then there is the chemical layer. Hair preserves a record of everything that entered the bloodstream during the period of the hair's growth. Because as the hair shaft forms, blood-borne substances become incorporated into the keratin matrix of the shaft. Drugs, toxins, environmental contaminants, medications, all of them leave measurable chemical signatures in the hair that can be detected through mass spectrometry analysis. A 90-day history. Every centimeter of hair growth represents approximately 1 month of biological time. The chemical analysis of a hair sample is not just a test for whether a substance was present, it is a timeline. It tells investigators not just that a substance was used, but when, with approximately 1 month resolution. And in the context of an investigation trying to build a picture of a suspect's lifestyle, habits, and recent history, a chemical timeline encoded in a hair shaft is the kind of background intelligence that cannot be obtained from DNA analysis alone. Let's talk about what CeCe Moore specifically said about this evidence, because her public statements, while brief, contain layers of meaning that the summary coverage of this case stripped out. She called the hair sample a Hail Mary. That phrase is precise and intentional coming from a scientist. A Hail Mary is not something you throw when you have other options. It is something you throw when the standard plays have been exhausted and you need the one improbable connection that changes the outcome. CeCe Moore, who has worked on hundreds of cases, used that specific language about this specific sample. She also said something that received almost no coverage in the subsequent days. She said that if the FBI lab received the evidence, they will get another chance, meaning another opportunity to derive a viable genealogical profile from a new source. Not a continuation of the glove work, not an extension of the existing CODI submission, a new chance. A second pathway to the same destination, a named suspect, through a different biological door. And she characterized the technical challenge specifically, the rootless hair, the complex mixture, the combination of those two difficulties in a single sample. She did not dismiss those challenges. She treated them as real obstacles that require advanced methodology to overcome. But she also did not describe them as insurmountable. Her public optimism, carefully hedged, professionally responsible, grounded in her understanding of what the current state of the art can actually do, was not the optimism of someone who did not understand the difficulty. It was the optimism of someone who understood both the difficulty and the specific tools available to address it. That distinction matters. When a scientist who has personally worked on the Golden State Killer case, who has seen IgG succeed in cases far older and more degraded than this one, calls something a Hail Mary, but still describes it with cautious optimism, She is telling you something specific about her assessment of the evidence's potential. Not a guarantee, not a done deal, but a genuine forensic pathway. One that requires the right lab, the right methodology, the right computational tools, and enough viable biological material in the sample to survive the analysis process. The FBI lab has the lab. Quantico has the methodology. The mixture deconvolution software was expedited specifically for this case. And the hair was sent to Quantico, confirmed by NewsNation's Brian Enton, by the Fox News DNA reporting, and by the ABC News confirmation of advanced new technology deployment. Everything that CeCe Moore said you would need, the investigation now has. Everything that needs to be in place for the Hail Mary to have a chance of connecting is in place. Here's where the story of this specific hair becomes part of a larger story about what forensic science can do in 2026 that it could not do in 2020, 2015, or 2010. The Darrell Johnson case took 40 years to solve, not because the investigators were not trying, but because the technology to analyze that specific hair evidence in that specific way did not exist for most of those 40 years. The hair was in the freezer, the case was open, the investigators never stopped, but the tool they needed, the whole genome sequencing approach applied to degraded rootless hair, had to be invented before it could be used. It was invented. Dr. Green invented it. Estrella Forensics operationalized it. The Darrell Johnson case proved it works. And now it is available. Not as an experimental technique in a university lab, but as a deployable forensic service that can be engaged by law enforcement on active cases. The Bear Brook case in New Hampshire. 4 victims, 50-gallon drums, decades unidentified, was cracked using hair from one of the victims when all other DNA sources had been exhausted. The hair produced a genealogical profile. Barbara Ray Venter built the family tree. The victims were identified. That case happened before the Darrell Johnson case, which means the hair to genealogy pipeline was working for victim identification before it was applied to suspect identification. The Nancy Guthrie case is asking the same technology to do the same thing from the suspect side of the evidence. The key insight from both of those cases is this. The technology does not care about the age of the hair or the complexity of the analysis, as long as sufficient biological material survives the extraction and sequencing process. The limiting factor is not the technique's capability. It is the quantity and quality of surviving DNA fragments in the specific sample being analyzed. And that is why the investigators who collected evidence from Nancy Guthrie's home and who ultimately sent the hair sample to Quantico rather than relying solely on the Florida private lab's analysis, made a decision that has the potential to be the most consequential forensic decision in this entire investigation. Let's go deeper into what the FBI lab at Quantico specifically does differently from a private contractor. Because the public discussion of this case has treated the routing decision, Florida versus Quantico, as primarily a political or institutional story. It is also a technical story, and the technical dimension is more important. The FBI laboratory has approximately 500 scientific experts working across divisions that include DNA analysis, trace evidence, chemistry, digital evidence, explosives, and forensic audio and video. Within the DNA unit, the laboratory has validated and operationally deployed next-generation sequencing using the MiSeq FGX system, the first NGS platform specifically validated for forensic casework and approved for the FBI's National DNA Index system. That validation means the profiles produced by that system are court admissible in a way that profiles from non-validated platforms are not. The FBI lab also has formal relationships with private forensic genealogy companies, including access to the methodological pipeline through which unknown profiles can be submitted to genealogy databases for comparison. Othram in Texas, Estrella Forensics in California. These are companies whose tools the FBI's investigative structure can access through formal, legal, and institutional channels that private county labs do not have. And the FBI lab has something that no private contractor can offer: institutional memory. Hundreds of cases, years of comparative data, peer-reviewed, published validation studies, and the specific forensic experience of having processed similar evidence in similar cases across decades of law enforcement partnership. When an analyst at Quantico looks at a rootless hair from a crime scene, they are drawing on a library of comparative knowledge that does not exist in the same depth at a private lab in Deerfield Beach, Florida, that primarily handles standard submission work. That institutional depth is what CeCe Moore was signaling when she said the FBI would get another chance with the hair. She was not just describing a new sample. She was describing a new institutional context, one in which the evidence is being processed by the right people, with the right tools, in the right building, with the right forensic memory, and the right analytical pipeline to pursue every biological dimension of what the hair contains. Now let's talk about the timeline of what is happening right now. Because understanding the sequencing of events, what Quantico is doing in what order, and what each step reveals before the next step begins, is the key to understanding why this evidence has not yet produced a public result, and what the path to a public result looks like. Step 1. Physical morphology examination. Every hair analysis begins here. Is the hair human? What body region did it originate from? What is the racial origin of the donor? Was the hair forcibly removed or naturally shed? Is there any visible follicular tissue? Are there any chemical treatment residues? This step takes days rather than weeks and produces the first informative data about the unknown contributor. Step 2. DNA extraction. Specialized extraction protocols for rootless hair use techniques derived from ancient DNA methodology. Silica-based extraction buffers, low-volume reactions, extended digestion conditions that are optimized to recover the maximum possible quantity of the degraded, fragmented DNA present in the hair shaft. This is where Astrea Forensics and Dr. Green's innovations come in. The extraction is the most critical step because it determines how much biological material is available for everything that follows. Step 3. Library preparation and whole genome sequencing. The extracted DNA fragments, short, degraded, but present, are processed into a sequencing library. Adapter sequences are attached to the ends of the fragments, allowing them to be read by the sequencing platform. The library is then run on a whole genome sequencing instrument, producing hundreds of millions of short reads that collectively cover the genome at approximately one-fold average depth. This step takes days to weeks depending on the priority of the case, the sequencing depth required, and the instrument availability at the laboratory. Step 4. Bioinformatic processing. The raw sequence reads are processed computationally, aligned to a reference genome, variant positions identified, genotypes called across the genome. This step requires significant computational resources and produces the genotype file that is the foundation for everything that follows. Step 5. Genealogy database comparison. The genotype file, formatted appropriately, is uploaded to a genealogy database, GEDmatch, Family Tree DNA, or another platform with forensic access agreements. The database returns a list of biological matches, people who share DNA with the unknown profile at a level consistent with a known genealogical relationship. Step 6. Family tree construction. A forensic genealogist, someone with a specific skill set to build family trees backward from a common ancestor and forward to a living candidate, takes the database matches and constructs the biological family tree that connects those matches to the unknown contributor. This is the labor-intensive step. It requires genealogical records, historical documents, death records, census data, and the specific navigational expertise to distinguish between the branches of a tree that lead toward the suspect and those that lead away. Step 7. Candidate identification. When the tree resolves to a living person who matches the age, gender, geography, and physical profile of the suspect, that person becomes the genealogical candidate. Step 8. Confirmatory sample collection. Investigators surveil the candidate. A discarded biological sample, a coffee cup, a water bottle, a cigarette, is collected without the candidate's knowledge. The sample is tested against the unknown profile. Step 9. Match or no match. If the discard sample matches the unknown profile, the genealogical hypothesis is confirmed. The investigation has a name. The warrants follow. The arrests follow. The prosecution follows. That is 9 steps. Some of them take days. Some take weeks. Some, particularly the family tree construction for a complex case, can take months. The Quantico hair submission happened in mid-April. It has now been approximately 7 weeks since the FBI confirmed receiving the hair DNA data. 7 weeks. In the timeline of an IgG investigation involving a complex mixture and a rootless hair sample is consistent with the earlier steps being complete and the later steps, tree construction, candidate identification, being in active progress. Thank you for watching this video. Don't forget to subscribe to this channel.